Hello students, very good afternoon. So before we start the session, I'm going to launch a technical poll. So please let me know if everything is all right. Please try to, yeah. Is the teacher audible clearly? No. Is the blackboard visible? Okay. So give me a second. Is the audio okay now? So I'll relaunch the poll. Okay. Give me a second. So now is it audible? No. Okay. So can you just try to put it in the chat box? Like if my voice is very low or is it not audible at all? First, you just help me with that. Please enter the please tell the problem in the chat box. No, yeah, we have here. So sound is not audible. Give me a second. Okay, wait. Hello. Guys, can you reconfirm about the audio? Is it still not audible? Yes, no, it's 50-50%. So 3 people are telling no, 2 are telling no. Okay, wait. Now is my voice audible? What is happening? Hold on, people. We launch. Now just tell me now. Is my voice clear now? No. How is it even possible? For everybody, it's like no. Is it okay now? The rest 2 of you, or can you please try to write it in the chat box, people? Like, why is my voice low or completely not audible? That I'm not able to understand. Sound is not audible. Yes. Is the sound audible clearly? Some are telling yes and some are telling no. So fine. So what I'll do is I will try to teach for 5 minutes. Okay. And then we'll try to relaunch the poll again. So if still there is some audio problem, you just let me know. Okay. So hold on. Not audible. So it's for one girl, right? Esmina. What about others? JNV. We have few people. We can ask the help. Okay. So JNV, Surendra or Alima, can you please confirm how is the audio for you? Make it fast, people, please. Okay.
Sir, now it's audible.
Thank you. Finally. Okay, people. Today we have to start. Our topic with photosynthesis. So we are going to have a revision session. So where we will talk about a certain extent. Half of the chapter will be finished today, and half we will take it in the next session. Okay. So when we talk about photosynthesis, So why do we need to learn about photosynthesis? That will be our first question. So when we talk about photosynthesis, we are understanding two basic things that that are very important for the life to exist on this planet. One thing is the energy, and the second thing is the oxygen. So if you see with respect to energy, the plants are the primary producers of the energy on our planet, because without plants there would be no energy for any of the ecosystems to exist. Okay, you take herbivores or carnivore or omnivore, any of these organisms who are heterotrophic, it is impossible for them to survive without the presence of plants. So plants have this exceptional capability of harvesting the sunlight. Okay, and then one second, people. So we have audio problems still. What to do? Okay, wait. I'll do one more thing. Students, I'll relaunch the poll. Okay, I'm sorry about it. I'll relaunch the poll. So we'll take the majority. Let's see if we have the same technical problem persisting. Okay. Okay.
Sir, before it was mine problem also. Then I have clicked that join audio option. Then it's become okay.
Now the problem is solved, right? So it's actually kind of, bothering me because without audio there is no class, right? So that's why I'm trying to fix this problem as soon as possible. So now we are good, so it's fine, we can move on with the chapter. Okay, so we were talking about the importance of photosynthesis, and I was telling plants are the primary producers of this energy. So they have the capability of harvesting this light and fixing it in the form of energy sources like glucose or any of the carbon based energies. Okay, but other reason why we study photosynthesis is because of the production of oxygen. Okay, so we understand the importance of oxygen, right? So all of us are dependent upon oxygen for our respiratory processes, and it comes majoritarily because of the photosynthetic organisms. So let us go back to the history a little bit and understand how much of ideas and how many different contributions led to the complete understanding of photosynthesis as we understand today. So like how we understand photosynthesis and the energy cycle of these plants, it did not start from the beginning with the same understanding. So it has taken some years of research and years of several people's contribution to give us the ability to understand photosynthesis. So the historical perspective of photosynthesis began in early, it's been for a very long time, but scientifically it started by 1770s by Joseph, Priestley. So he was very brilliant because he conducts this experiment called as, candle with jar bell. Okay, you can bell jar. So in this experiment, what he does is he will try to take a bell jar. Okay, bell jar would be like this, right? Like a dome shaped, glass thing. So this is called as bell jar. So what he does is he will try to place a candle, and then it's completely sealed, there is no exchange of air. So the candle would die off very soon because there is no gaseous exchange. And same goes with mice would suffocate very fastly when the jar was enclosed. And then he goes on try to put a plant where he uses mint plant, and when he puts a mint plant, what happens here is the lifespan of this candle as well as the suffocation of this mice which was in the bell jar was not seen. Okay, it was the lifespan of this candle was extended. And when you see with respect to the suffocation of mice was not suffocating so much, or it was not suffocating at all when the plant was there at that time. That gave an idea for Joseph to conclude that, okay, so the plants have the capability of taking up carbon dioxide and then it releases oxygen. Okay, so then later on what happens, using the similar ideas that were set up by Joseph, okay, Eganhouse in 1779, what he did was, the oxygen release was only possible in sunlight, in the presence of sunlight. Only when there is sunlight the plants go and undergo this process where there is production of oxygen, he discovers. Okay, So he was able to distinguish that whether it is happening in the light conditions or dark conditions. And later on, what happens? Theodore comes. In Theodore, what he says that for everything to take place, for the plants to even undergo photosynthesis, water is very essential, he will conclude. So now he was not able to prove how water is useful for the photosynthesis, but he observed that in the absence of water, the plants dried up and they did not produce any photosynthetic byproducts, like there was no production of oxygen in the later cases. Next, what happens? Julius Vaughan comes and he says that green plants are able to harvest this harvest this energy, and they are able to do it, okay, by producing glucose. Okay, they produce glucose, and they're also stored in the form of starch. That is what he concludes. Okay, then Eagleman, this is one of the most brilliant experiments that is conducted by a scientist to understand what type of light is used for the photosynthesis process. So here what he does is he will try to take the visible spectrum, that is our VIBGYOR in our light, and he will try to study the photosynthesis, what is the range of photosynthesis or rate of photosynthesis with respect to different wavelengths of light, like violet, blue, green, yellow. So as he was studying, he understood that there is specific spectrum that is used for the photosynthesis, not the entire light is used for the photosynthesis. Okay, so we will call it as photoactive radiation, so which is used for the photosynthesis process. So that is what he proves. Now what happens? Von Neal, he actually he suggests that he will give an empirical formula for the process of photosynthesis. Here what he tells is essentially light is dependent upon photosynthesis is dependent upon light, and hydrogen is hydrogen is obtained by oxidizable compounds. For example, if you take in normal plants, we take water. So here water is oxidizable and it is broken down into hydrogen and oxygen, right? So we have hydrogen and oxygen when we when we break the water molecules. So this hydrogen that is being extracted, so that will result in the formation of electrons. So basically for the photosynthesis, what is required? A hydrogen is required, correct? No? And then that when that is extracted by oxidizable compounds, okay, so they gain electrons for the photosynthetic reactions. That is what he says. And after this idea that was put by him, so the derivation of empirical formula he gives was not only H2O, any of the oxidizable compounds, when it reacts with carbon dioxide in the presence of sunlight, okay, it will give rise to what? It will give rise to the hydrogen. Okay, for example, this is oxygen. Okay, so here we have the glucose, and then we have the water molecules as the byproducts. Okay, next, Hill proves that oxygen evolution only happens in the light reaction through his experiments. And finally, Kevin comes, and Kevin actually traced back the entire biochemical pathway of the photosynthesis. Okay, so these were the contributions that happened in the past. And finally, today we just try to summarize photosynthesis by trying to tell that, give me a second. Okay, by trying to tell that photosynthesis is a physico-chemical reaction. What is physico? Physico is like where our plants will use the sunlight to break the water molecules. Okay, and then we have chemical reactions. Chemical reactions is conversion of different products and finally reducing it into sugar molecules. Okay, so overall chemical reaction of photosynthesis would be 6 molecules of carbon dioxide is used in the presence of 12 molecules of water. So sunlight, chlorophyll, and enzymes, they participate in this reaction to give rise to glucose, that is C6H12O6, and then it eliminates or liberates 6 molecules of water and 6 molecules of oxygen. So this is the perfect understanding of chemical equation of photosynthesis that we have derived for now. So where does the photosynthesis take place? Correct? No, that is the first question. So if you see, where does the photosynthesis take place? Photosynthesis takes place in chlorophyll. So where are the chlorophyll present? Chlorophyll are abundantly present in mesophyll cells in the leaves. Okay, so the number of chlorophyll actually varies from species to species. So if we see the presence of chlorophyll in the mesophyll cells, you can see that they are organized in very close to the walls of this mesophyll cells. So if you have mesophyll cells, you can see that the chlorophyll is quite organized, okay, near the membranes of the mesophyll cells in layers. So why do they do this? Because they want to interact as much as possible with the incident light. Okay, how much of our light is that is being incident on this leaf, that is it is getting how much of light it is receiving or incident. So it wants to interact as much as possible. That is why it is organized at the borders or membranes of this mesophyll cells. Okay, so if we see chloroplast, what is chloroplast? We know that chloroplast has membranes, outer membrane and inner membrane, and they are separated by periplastid space, that what we call it as. And within this periplastid space, we have something called as stromal matrix. So within the stroma, there are your grana. The grana is organized, and they are also interconnected by stromal lamella. So the grana is made up of coin-like structures called as thylakoids, like this, no? So they are organized like thylakoids. So within the thylakoid membranes, what do we have? We have chlorophyll pigments. So when we look at chloroplast, chloroplast is not just made up of one type of pigment, it is made up of several type of pigments. Okay. So for example, chlorophyll A, okay, chlorophyll A, chlorophyll B, okay, carotenoids. Okay, the names of it, all the pigments is in the next slide. Carotenoids, xanthophyll, okay, xanthophyll. So these kind of pigments are present in the chlorophyll, and that is why our leaves and all, they appear in different colors. For example, they appear greenish, they appear light green, yellowish, brownish, orange. Okay, so based on the type of pigments that are more in the different species of plants, that is the color of the leaves we have. So why do we need different photosynthetic pigments? Okay, so our plants, they have the ability to utilize a specific wavelength of light. Okay, if you see in a very broad sense, but if you see very in detail, many of the accessory pigments like chlorophyll B, xanthophyll, and carotenoids, what they're doing is they're helping the plants to receive other wavelengths of light also, so that it will help the process of photosynthesis. Now in chloroplast, what is happening? As I said, in chloroplast, we have three important regions. What is that? First one is your grana or your thylakoids. And the second one is your stromal lamellae. And the third one is the stroma. Okay. So in these three regions, the photosynthetic reactions are happening. So if you take your grana and if you take the stromal lamellae, here what is happening? The light reactions of photosynthesis is taking place. But if you consider the stroma, in the stroma, what is happening? The stroma, okay, they have specific enzymes and proteins where the dark reactions of the photosynthesis will take place. Stroma is the region of the biosynthetic process of photosynthesis will take place. Okay, biosynthesis process. Whereas here we have light reactions when it comes to grana or stromal lamellae. So this is the structure of chloroplast and the functions of the chloroplast. So let us see the properties of your photosynthetic pigments. So the question is, as I said, at what wavelengths or at what particular light fractions is our photosynthetic pigments very active, and the photosynthetic rate is high. Okay, so if you take chlorophyll A, okay, if you take chlorophyll A, so if you observe the wavelengths, right? So chlorophyll A is very active at the wavelengths between red and blue. Okay, so they're very active between red and blue. Similarly, when you see chlorophyll B and chlorophyll B, carotenoids and xanthophylls, so they are okay with lower wavelengths of light. But for the chlorophyll A, it requires minimum of 680 to 700 nanometers of nanometer wavelength of light. Okay, so that is usually between red and blue. So whenever we understand the photosynthetic rate with respect to the wavelengths, okay, here you can clearly understand what is the absorption capacity. For example, if you see rate of photosynthesis and with respect to absorption, so you can see here when there is 700 nanometer, correct? No, when there is 700 nanometer, you can see that there is high amount of photosynthetic byproduct. There is high rate of photosynthesis. Whereas if you see in some regions, that is the wavelength between 400 to 500 nanometers, because of your accessory pigments like carotenoids and chlorophyll B and xanthophylls, they are able to observe even the low wavelengths of light. Okay, at these two positions, there is significant amount of what do you call photosynthetic rate. Whereas the intermediate wavelength between 500 to almost till 650. So there you cannot see any of the photosynthetic rate. You can see very minimal photosynthetic rate that is happening. So wavelength determines the rate of photosynthesis, and our pigments have specific wavelengths to which they become highly reactive. So this is about how wavelength is significant. So one more, we have an experiment called as immersion enhancement effect. So in immersion enhancement effect, what happens is he will take chlorella, which is an algae, which is again photosynthetic organism. So when he takes chlorella and he places in different lights, first what he will do, he will take both the lights, that is blue and red, and he will place the chlorella. Okay, when he ons the light, you can see that the rate of photosynthesis is very high. Then again, what he will do, he'll take one of the light and he'll on only red lights. Okay, so when he ons only red lights, there is only moderate amount of photosynthetic rate. When it comes to only the blue light, okay, far red, that is blue light, when you just look at it, again there is moderate amount of photosynthetic effect. So here what we can understand with respect to immersion effect is, when you talk about the photosystems, okay, or the regions where your light energy is used for something in photosynthesis, these are called as photosystems. So we have two photosystems, that is PS1 and PS2. So this immersion enhancement effect shows that individual photosystems, they have a capacity to harvest a specific wavelength of light, that is 680 nanometer is one photosystem and 700 nanometer is another system. Both the photosystems are involved in photosynthesis. That is how we understand when both of them are combined, you can see the photosynthetic rate. When individual is performing, you can see what is happening with respect to photosynthesis. Correct, right? So this was that. So coming to, okay, let us understand, let us try to understand more topics. Okay, before that, you will have to answer this question. So I'll launch an MCQ poll. Okay, so is the question visible? Wait, I will reduce this. I think so. Okay, fine. Anyways, I will launch it. Immersion enhancement effect and red drop have been instrumental in discovery of, what are the options we have? Photorespiration and cyclic, electron transport, oxidative phosphorylation. Third option is photophosphorylation and non-cyclic electron transport. And the fourth option we have is two photosystem operate simultaneously. So these are this thing. Okay, so one, okay, nice people. I think I will just move. Yeah. Try to answer, students, so that it's good. We can understand how the class is going on. If you're understanding the session or something like that, YouTube students can try to put it in the chat box. So you can write your answers in the chat box. Okay, be interactive. I'll be seeing you, right? YouTube students. So, all right, our time is up. It's more than 1 minute now. Oh yeah, I was waiting for the fourth person to answer. Anyways, 4 people have given 4 different answers. And what are the chances? Okay, so now I have ended the MCQ. Let us see what is happening. See, the immersions, just now we discussed about immersions enhancement effect using the red drops, right? So far red, and then you have red. When he used the two systems, I told you, when you see far red and you see red, the red has 700 nanometer in length, where 700 nanometer wavelength. When you see the far red, it is having around 680 nanometers. Okay, it's almost coming around blue. Now, when you see these both photosystems, let us go back to the prior slide before we answer this question. Just now we discussed, right? So whenever we have far red light and we have red light on, look at the photosynthesis. So that means at different wavelengths, what is happening? What do you tell? Your photosystems are behaving only to a certain extent. Okay, individual photosystems, see how they are behaving. But if when both the lights are on, see both the photosystems participate, and that is what gives rise to photosynthesis. You are understanding my point, right? If only one photosystem participates, no, then is only half of the photosynthetic byproduct will get, means the reaction photosynthetic rate is very, very less. Whereas when both the photosystems work simultaneously, so that is when you have more relative rate of photosynthesis compared to individual wavelengths. Did you understand? So the answer for this question is two photosystems operating simultaneously. Only when that particular wavelengths are available, both the photosystems operate. When both the photosystems operate, you have more photosynthetic byproduct. Okay. So anyways, don't worry. We will try to understand now in detail what we just talked about. So first we will talk about what is light reaction. Okay. So before we talk about light reaction, let us see what is your photosynthetic pigments, how they are arranged. If you look at the photosynthetic pigments, apart from your chlorophyll A, right? So apart from your chlorophyll A, we have other photosynthetic pigments like chlorophyll B, xanthophyll, and carotenoids, etc. Right? So these are called as accessory pigments, and these accessory pigments, okay, they all they are arranged in a very proper manner. Okay, okay, they are arranged in a very uniform manner so that they receive as much light as possible to their specific wavelength. So they form a complex called as light harvesting complex. So in light harvesting complex, there is every photosynthetic pigment except, okay, except what we don't have chlorophyll A. Okay, why don't we have chlorophyll A? So apart from LHC plus you have chlorophyll A. Okay, chlorophyll A, what it happens, the chlorophyll A will not be there. So this one molecule of chlorophyll A that it will be present in the photosystem, that is called that will create something called as reaction center. Okay, reaction center. I'll repeat again. So what I was saying is, apart from chlorophyll A, all the accessory pigments combine together to form some complex called as light harvesting complex. Whereas the chlorophyll A that is present in your light harvesting complex, okay. So here what is happening? This chlorophyll A will form something called as reaction center. Did you understand? So this chlorophyll A has the capacity of harvesting two different wavelengths of light. One is at 680 nanometer and other one is at 700 nanometer. So this 680 nanometer and 700 nanometer, we call it as photosystem, okay, PS 680, or we'll call it as PS 700. So these photosystems are called as photosystem 1 and photosystem 2. Now what is photosystem 1 for us? Photosystem 1 is 700 nanometer and photosystem 2 is 680 nanometer. Okay, what does this nanometer signify? At what wavelengths our chlorophyll A can harvest this energy? That is called as that signifies this wavelengths. Now this 1 and 2, does it signify their order of reaction? Like 1 is participating first and photosystem 2 is participating next? It's not like that. Okay, so this was just like based on discovery. People started discovering photosystem 1 first, and later they started discovering photosystem 2. That is why it is like PS1 and PS2. But when it comes to light reactions, okay, let us understand light reactions very, very clearly here. So what is happening in light reactions? First we will take photosystem 2. What is happening in photosystem 2? We have P680, isn't it? So this P680, what it does is, see, you have your chlorophyll molecule, okay, or pigment. Here light energy incidents on your photosystem 2. As the light energy incidents on your photosystem 2, this energy, light, is used for breaking down of water molecule. When a water molecule is broken down, what is it is giving? It is giving rise to hydrogen, it is giving rise to oxygen, and it is giving rise to electrons. Did you understand? So once this lysis of water happens with the presence of light, this is called as photolysis. Okay, so this reaction is called as photolysis. So once they have the photolysis, now why the photolysis? Because we wanted electrons. So once the light energy incidents, now what happens? The electrons that are present in your photosystem's atoms, they get excited and they just propel to their outer orbits. So once they are put out of the orbits, now they get excited, right? So as the electrons are getting excited, so these electrons are channeled through, okay, a transport mechanism. Okay, this is called as electron transport chain. But as the electron is moved, okay, moved from the photosystem, how does it move? How is the electron moved? Electron is moved down the gradient. So what kind of gradient are we talking about? We call this gradient as redox potential. Okay, hold on, give me a second. This gradient is called as redox potential. Okay, so redox potential, okay, it goes down the redox potential gradient. So as it goes up, okay, when it gets excited out of the photosystem, P680, and then what happens? Electron is transferred, electron is transferred down its redox potential, okay, to reach P700, okay. During this process of transferring of electrons, okay, this chain is called as electron transport chain. If you see electron transport chain is made up of series of proteins. Okay, so the series of proteins, what they're helping, this is helping in transfer of electrons from P680 all the way till P700. But here the molecule that is used for the transfer of electron is of low redox potential. Did you understand? So it goes down the gradient. As it goes down the gradient, what is helping for our electrons to get transferred? It is our there is something called as cytochromes. Okay, I'll write it here. There is something called as cytochromes. So what are cytochromes? Cytochromes are the specialized complexes which help in the transfer of electrons. Okay, they create something called as electron transport chain. So during this transfer of electrons, now what is happening? One molecule of ADP, okay, is from the free phosphate, it is added, and it will generate what? One ATP molecule is generated out of this reaction. When electron is moved from P680 to P700 using cytochromes, what is happening here? There is a chemical change where that results in the formation of ADP into phosphate. Did you understand? So once this ATP is created, now what happens? These electrons are successfully transferred to the photosystem 2. You can see here. Okay. So once these electrons are transferred to the photosystem 2, again the light incidence on your photosystem 1. Once the light incidence on your photosystem 1, which is capable of harvesting P700, again the electrons get excited. Once the electrons get excited, here what happens? Look here, people. Here the electrons is coming from photosystem 1 by it itself is generating these electrons, whereas when it comes to photosystem 2, you can see that the splitting of water gave rise to 2 electrons. So that is a very important difference when it comes to photosystem 2 and photosystem 1. So once the electrons that have been transferred from 680 to 700 comes here, and after the light, again these electrons get excited. Now this time when it gets excited, from the P700, now what happens? These electrons are again transferred down the gradient. Here for this time, a very high redox potential molecule is used for transferring of electrons down the gradient. So as the electrons are transferred down the gradient, that will result in, okay, see, NADP plus H plus, that will result in the formation of NADPH. Okay, as the electrons are moved down the gradient using higher redox potential molecule, now the hydrogen molecules which are in the lumen reacts with the NADP plus. Okay, so NADP plus is reduced to NADPH, okay, by the end of your photosystem 1. So this reaction, okay, from here to here, from here to here, okay, it resembles the shape of Z. No, that is why this this reaction is also called as Z scheme. Okay, the photosystem 1 and photosystem light reaction is also called as Z scheme. So what are the byproducts of your Z scheme? So the byproducts of your Z scheme or your light reaction is ATP and NADPH. So this ATP and NADPH that are produced by your light reactions, they are used in dark reactions, okay, for the further synthesis of your glucose. Okay, this is only half of a reaction. So if we observe these photosystems, so there are two things happening here. One is cyclic photophosphorylation, other one is non-cyclic photophosphorylation. So what is phosphorylation? Phosphorylation is a process of production of ATP molecules, okay, through ADP. See, what is phosphorylation? This is ADP. We phosphorylate ADP by adding another free phosphate to give rise to ATP. So this is called as phosphorylation reaction. So in phosphorylation, there are two types of phosphorylation. What is that? One is cyclic photophosphorylation. Cyclic means it is happening in a cycle. Photo means light. Okay, phosphorylation means I told the definition. So one is happening in a cyclic fashion, other one is happening in a non-cyclic fashion. What is happening in cyclic fashion? The conversion of ADP into ADP plus P into ATP. So this ATP production is happening in a cyclic manner. So let us see how does it happen. If you take photosystem 1, so where is photosystem 1 present? That is very, very important. Photosystem 1 is present in stromal lamella. So since it is present in stromal lamella, there is no photosystem 2 in stromal lamella, and there is no NADP. Okay. Sorry, they do not have the enzymes called as NADP reductase. Okay, they do not have the enzyme called as NADP reductase. So that is what is photosystem's location is about. Okay, so now what happens? The photosystem 1, when it receives light, see, there is no water here, right? Correct? No, there is no water that is being split using the light. So when the photon incidence on the photosystem 1, now what is happening? From your photosystem 2, the electrons were transferred to the photosystem 1. Simultaneously, when the light falls, the electrons from your photosystem 1 gets excited. Once they get excited, now what happens? They are they're moved down the potential. As they're moved down the potential, now what is happening? The electrons are used. Okay, see here, ADP is creating ATP, again ADP is creating ATP. So here, see here, the electrons are being chained, okay, through electron transport chain. And again, what is happening? After moving through several pigments, after can you please mute? Right. Yeah, let us go back here. So as I was saying, see, as it is moved by, see, pyridoxine, okay, cytochrome, I told you what is cytochrome B6, okay, again cytochrome F or plastocyanin. So this is how the electrons are moved down the redox potential. So here it is higher redox, pyridoxine is having higher redox potential as the electrons are moved, okay. So that will result in changes in the system that helps in the formation of ATP, okay. So since the production of ATP happens with the help of electrons, and the electrons are derived by the photosystem 1 itself, independent of water, okay, here the cycle of electrons happens continuously, okay. That is why we call it as cyclic photophosphorylation, okay. When it comes to non-cyclic photophosphorylation, when it comes to non-cyclic, what happens? We discussed about here, non-cyclic. You can see here, for the electrons, the depending factor for the electrons for the photosystem 2 is what? Water molecules. Only when there is water molecules, the electrons are derived. When the electrons are derived, okay, then the electrons are transferred down the chain to produce this ATP molecule. Correct? No, people? So here, the flow of electrons does not take place in cyclic manner. That is why we'll call it as non-cyclic photophosphorylation. So this is cyclic photophosphorylation. Okay. So what is the significant difference between cyclic and non-cyclic photophosphorylation? Very, very important. Okay. See here, in cyclic photophosphorylation, PS1 is involved. In non-cyclic, both PS1 and PS2 are involved. Here I told you, as I told you, in cyclic, water is not required. Here, photolysis of water is required for getting the electrons. Next one, oxygen is not evolved in cyclic. Oxygen is evolved in non-cyclic. NADPH is not synthesized in cyclic because it does not have the enzyme called as NADP reductase. Okay, whereas in non-cyclic we have both. We have NADP is synthesized because it contains the enzyme that is essential for this purpose. Next one is ATP. See, for example, used to produce additional ATP in order to meet energy demands, whereas here products can use products can be used for the light independent reactions. So for either of the cases, what happens? For the production of ATP, when the cell have an emergency for ATP, it will go for cyclic photophosphorylation, or if it has to go for light independent reactions, so it will go for non-cyclic photophosphorylation. So this is a significant difference between these two light reactions. Okay, so fine, we have another question here, students. So I will launch a poll again. Okay, continue. 1 second. Yeah, so here's your poll. Okay, yeah. During the light reaction of photosynthesis, which of the following phenomena is observed during cyclic photophosphorylation as well as non-cyclic photophosphorylation? Release of oxygen, formation of ATP, okay, formation of NADPH, and involvement of, okay, PS1 and PS2 photosystems. Okay, 40 seconds. You have 20 more seconds to answer. Okay, I will give you another 5 seconds extra. All right. How many students have answered? So we have good participants now. I need 2 more. Okay, 3 more people needs to answer. Okay. Come on, people, time up. One more. Do you have one more answer? Okay, can we continue? Okay, okay, good. So we have A, C, and D as your options. Okay. I think we have to read the question twice because we have multiple answers. That means, in 3 of you have answered, 2 are wrong. Okay. Or maybe 3 are also wrong. Let's see the question properly. During light reaction, okay, during light reaction of photosynthesis, which of the phenomena is observed during cyclic photophosphorylation as well as non-cyclic photophosphorylation? In both the cases, what is the one thing that is common there? Talking with respect to light reaction, they're asking. Okay, release of oxygen and formation of ATP, okay, formation of NADPH, and involvement of photosystem 1 and photosystem 2 pigment systems. Okay, listen here, both have to be having the common factorial, right, people? So when we talk about photosystem 1, was there for release of oxygen? See, come back here. If you come back here, it is clearly told here, oxygen is not evolved in photosystem 1 when it goes to cyclic photophosphorylation. So that is wrong. Again, come here. Formation of NADPH. When it comes to formation of NADPH, look here, NADPH is not synthesized. In which one? Cyclic photophosphorylation. Okay, let us come to formation of ATP. Okay, which is another option. When it comes to formation of ATP, look here, people, we have cyclic and we have non-cyclic, correct? No, so we have cyclic and we have non-cyclic. In both the cases, what is happening? We have ATP here, we have ATP here, correct? No, so when we have ATP in both the case, this is the common phenomena that we can observe in the photosystems, isn't it? So this is correct. So why not this is not correct? Involvement of PS1 and PS2 pigments in the see, for example, if you take photosystem 1, pigments are different. I told, no, the pigments are quite different in both the photosystems. But when it comes to cyclic or non-cyclic, in non-cyclic both the PS1 and PS2 are used. When it comes to cyclic, only PS1 is used. So it's not common, no? So that is why the 4th option is wrong. So the correct option is formation of ATP. Formation of ATP is happening in both the cyclic as well as non-cyclic photophosphorylation when it comes to light reaction. Okay, sorry. So we will discuss very important topic now, and it is also very interesting to learn about this topic. So this is called as Chemiosmotic Hypothesis. So what is Chemiosmotic Hypothesis? So one of the important concepts that we have to understand before learning this is, when we look at living organisms, all our living organisms, we have a capacity to store energy in the form of chemicals, right? So what do? We will try to, create certain, certain molecules like ATP, where when you try to break the bonds between, when you try to reduce the ATP, so this breaking of bonds, like converting ATP into ADP plus phosphate, right? When you do that, the energy is released. So we try to, we try to make an energy source by creating chemical bonds, okay, and we store these chemicals within us so that whenever there is a requirement, we can break these bonds and derive this energy. So this is one of the very unique things that is happening. So this is called as what? So when you see your mitochondria, right? So what is happening in mitochondria, people? In mitochondria also there is ATP synthesis, isn't it? Correct, no? So this is called as phosphorylation is happening. So whenever we talk about phosphorylation in mitochondria, so mitochondria will produce ATP, and for the production of ATP, what it uses is, it uses something called as proton gradient, correct, no? It will use something called as proton gradient. So the mitochondria uses this proton gradient for the synthesis of this ATP, whereas the same mechanism happens even in plant cells apart from mitochondria, that is with respect to your chlorophyll, okay, chloroplast. When it comes to chloroplast, especially the production of the ATP is happening in thylakoids only. Why we are talking about thylakoids? Because your pigments are present in thylakoids, correct, no? So when we see the structure of thylakoids, the thylakoids have a membrane, okay? The thylakoids have a membrane, and within the membrane, they have different pigments, okay? They form the photosystem 1, photosystem 2, and they're also having, electron transport chain mechanism, and then, or electron acceptors, we can tell. And then they also have the, a certain proton pumps. So when you see thylakoids, the thylakoids is also having something called as quantasomes, where the light reaction is happening. Okay, so that is why thylakoid structure is very, very important for us to understand the ATP synthesis. Okay, so here thylakoid, what is happening is, let us compare between mitochondria and thylakoid. If you see mitochondria with respect to your respiration chapter, here what is happening in mitochondria? For the production of ATP, a proton gradient is generated. When you see where is the proton gradient established, if this is your mitochondria, this is your inner membrane, and this is the outer membrane of the mitochondria. Okay, so between the two membranes, okay, in the intermembrane space of the mitochondria, what is established? A proton gradient is established, which further which further is responsible for the production of ATP. But when it comes to thylakoids, what is happening here? The proton gradient is established in the lumen region. Okay, see here it is given high H plus. That means we are not trying to create a proton gradient in the intermembrane space in thylakoid, but thylakoids have different mechanism where the proton gradient is created within the lumen of the thylakoid. Now let us see some of the important aspects of some of the important aspects of why the proton gradient has to be established first, and then what happens with the establishment of proton gradient and how it finally results in the formation of ATP molecules. So let us go one by one. First, we have a thylakoid membrane, and within the thylakoid membrane, we have our photosystem 2. So in photosystem 2, we know what is happening. The light incidence. When the light incidence, what is happening? Our P680, it uses the light and it breaks down water molecule into O2 and H plus. Correct, no? O2 and H plus, and the electrons are released. Can you see here? So the electrons are released. So when the electrons are released, here what do we have? Oxygen, what it does? It will diffuse out of the thylakoid, out of the stroma, and it is removed from the plant in the form of respiratory, like through stomata. Whereas when you talk about your H plus ions, the H plus ions are where are they present now? Lumen, correct, no? So the H plus ion concentration keeps increasing in the lumen by photolysis reaction that is done. Next one, what happens? The electrons that are released have to undergo, okay, a transfer. Electron transfer has to happen. For this electron transfer, who is responsible here? ETC, electron transport chain, is responsible. So the electron during the electron transport chain, okay, these molecules like the PQ or PQH, what is given, these molecules, they use a proton, okay, they use a proton. Here the proton that was used for the transfer of electrons from 680 all the way till P700, okay, was a simple hydrogen molecule. So now what do? They take the hydrogen molecule which is already present in the stroma. See, don't confuse. This is your chloroplast. Okay, so this is your okay, grana, and the coin-like structures are called as thylakoids, right? So this is called as stromal lamella, like this, correct? No, people. So here, whatever this space is there, this is called as stroma. Okay, whatever we are talking about here, these coin-like structures are called as thylakoids. Okay, now you know what we are talking about. Whatever this region we are talking about here, there were hydrogen ions, and these hydrogen ions, what they are pulled inside. Why they are pulled inside the lumen? Because for the transport of electron across the membrane, a proton is moved, okay, across the membrane, so that electron transfer takes place from one photosystem to another photosystem. Did you get it? Now again the concentration of hydrogen increased. Now let us see what happens. Oh, 350. I think I will try to just finish this. Okay, I think till 4 is the class. Okay, so next what happens after the electron transport chain? After the electron is transported to the photosystem 1, correct? No, so hydrogen concentration again increases in the stroma, in the lumen of the thylakoids. Next, what is happening in the photosystem 1? Okay, outside the in photosystem 1, what is happening here in PS1? NADP plus is reduced to NADPH using an enzyme called as NADP reductase, which is present in the stroma. Okay, so in the stromal matrix, this enzyme is present. Okay, so here what happens, additional hydrogen ions from the stroma is taken up for the reduction of NADP plus to NADPH. Okay, so again the hydrogen concentration decreases in the stroma. Now since there was movement of hydrogen into the lumen so much, now what happens, the proton gradient, okay, a proton gradient is established within the lumen of the thylakoid. Understood? So when the proton gradient is established, now what happens, the more and more proton gradient keeps on increasing, because as the protons are moved across the membrane, the more proton gradient is established. Okay, now this increased proton gradient in the lumen will result in one thing. Okay, it will change something in the membrane of the thylakoid. What is it changing? If you see here, there is in the membrane of the thylakoid, okay, there are ATP synthetase, which is an enzyme. Okay, the ATP synthetase enzyme has two components. One is CF0 and CF. Okay, they have two components. So these ATP synthetase component, what we are talking about, they are proton pumps. Okay, so what is happening, this CF0, because of the hydrogen concentration, when the hydrogen ion concentration increases, now this CF0 facilitated diffusion. Okay, using facilitated diffusion mechanism, they transport this hydrogen ion outside of the thylakoid. Did you understand? So this is facilitated diffusion. And this is happening as the CF0, what it is doing, it is taking the protons and it is leaving into the stroma. So this is indirectly their proton pumps. Okay, when the proton pumps does this, that is CF0 does this thing, now what happens, that will induce a conformational change in the other component of your ATP synthetase, that is CF1. The CF1 undergoes conformational changes. Okay, why it undergoes conformational changes? As the proton gradient was increasing, pH was also changed. Okay, and then electron gradient also changed. And then now what is happening, as the protons start moving outside, the conformational changes in the ATP synthetase CF1. So when that happens, that will result in, okay, ADP plus phosphate. ATP synthetase, what it will do, it will take adenosine diphosphate and it will take free phosphate molecules in the stroma and it will combine them to produce more and more of ATP. Okay, so this pathway, okay, this cyclic reaction that is happening, okay, and the establishment of proton gradient, okay, the role of protein pumps and the presence of ATP synthetases will finally result in, okay, the formation of ATP molecules. So this is about chemiosmotic hypothesis, okay, for the production of ATP that happens in thylakoids. Okay, so now we have finished your light reactions. We have dark reactions and photorespiration that we will continue in next class. Is it clear, students? Okay, so before we end the class, I will give you a poll. Okay, NPS poll. Please try to answer this. Is it launched? Yeah. So do it as soon as possible, students. Okay, let's see. Be free to fill an NPS form, okay? Because it's always to help you to improvise the teaching. So it should be helpful if you answer it, because we will know where the things are not happening, so that it will be fixed in the next class and you might fill the classes back again. Okay, and it becomes interesting. Make it fast. We have 5 questions. Okay.
Sir, I'm not getting the poll.
You're not getting the poll? I think there is poor network. Okay. See, okay, this is about, you know what is NPS form probably by now. So if you have any problems with the way, okay, if you're not understood any things, so you can just put it in the message, so that will also be helpful. Okay. Okay, students, I will end the poll. Okay, this should be enough. All right. Yeah, thank you for very much for your suggestions, and we can continue the next class. Okay. As I keep saying, it's all right with anything, but when it comes to doubts, or if you have any topic that you have not understood, or if you felt that it just slipped, you cannot grab it, or grasp what I was trying to tell, you can just put it in the chat box. Okay, so it will take a minute or two for us to revise the topic and talk about it in a very simplistic manner. Okay, so that will help you to understand. Apart from that, everything is fine. Okay, so you'll gradually pick up with the class. Anyways, guys, thank you very much. Okay, have a nice weekend. See you. Bye bye.
Thank you, sir.